right, so again, my name is Federico Lucchetti from the University Hospital of Lausanne. I'm going to show you how to construct the first MRSI metabolic-based connector. We already covered this, so we are working here with the hydrogen whole brain MRSI, not the phosphorus one. We are getting a 5mm isotropic resolution in about 12 minutes time. With the latest upgrades called eccentric, we can get to about 10 minutes. I'm not sure. Was it 10 minutes? Yeah. These are the 5 metabolites that we're seeing. We're seeing those because that's the only one that we are spectrally resolving. So we get on one hand myoinositol, choline, and acetyl aspartate, glutamine plus glutamate, which forms a complex called GLX, and then creatine. Okay, we more or less know what these metabolites mean at the cellular level. It's a little bit trickier to know what they mean once we measure them macroscopically in an MRI scanner. Nevertheless, we know that from neurological lesion studies that they are somehow involved in a much more complex network. They are not independently varying from each other. They have to be involved in some kind of balancing network that we would like to uncover. And the best way to do it, having at disposal these MRSI whole brain maps, we could simply just construct a macroscopic level metabolic connectome that would capture this interaction between these metabolites. Okay, for those of you who work in structural connectomics, we all know these principles, right? So the wiring economy of white matter neurons, usually what you do is neurons tend to minimize the wiring cost. Why? Because it's metabolic or energetically much more efficient. It takes so much energy to have these long-range connections. That's why we are preferentially wiring between neighboring gray matter regions. If we only wiring neighboring regions, we're getting high clustering coefficients. So one way to connect them, we're getting a maximized global efficiency, so very sparse long-range connections. And I was wondering whether we could formalize this economy of brain networks also in the metabolic case. Okay, what's the application of all of these things? Well, there are multiple ones, but the one I'm interested in, and the research group where I'm working at the Center for Psychiatric Neuroscience in Lausanne, is to find biomarkers for early onset of psychosis and schizophrenia. We would like to start first building foundational knowledge, ground our findings with biological ground truths, and then maybe come up with a framework, a comprehensive framework that clinicians can use to guide their diagnostic in the most comprehensive way. Once we have done this, we also want to answer a very important question when we are acquiring this data, is to do these disruptions in metabolic networks precede the structural ones. Because the structural ones, usually we can detect them only very late in the chronic stages of schizophrenia. Okay, let's get right into it. So to construct the network, we have to define the nodes and then the edges. For structural connectome, it's easy. We get access to the spatial dimension. The nodes are just the static gray matter regions. The edges are the fiber tracks. For functional, we have that extra temporal dimension. And now we can define the nodes are actually dynamical because we get multiple snapshots across time, and we're going to define the edges as a synchronicity measures between the gray matter, the dynamics of the gray matter regions, maybe with a Pearson correlation. For MRSI, it's a bit different. We only have access to space. Nodes are still static. We could then define the edges maybe as a correlation between those metabolic profiles and formalize it as a biochemical similarity. And that's actually what we do. We start first with an anatomical scan. We postulate it with your favorite atlas. We can also use the cubic postulation, which we did. And then you register that on your MRSI page with this lower resolution. Once you have done this, you're creating these vectors, which contain 5 average concentration of these 5 metabolites in that particular parcel. You did this across the brain, including the cerebellum and the subcortical regions. You compute all the cross correlations, and you come up with this matrix. Red means positive correlations, blue means negative correlations. We can do this for one single subject. We can do this for multiple subjects, where we scanned about 68 healthy adolescents between the age of 13 and 15 as a pilot study. Or you can just average all of them together to get this matrix. You can see the modular structure appearing here. So here are the neocortical hemispheres, left and right. Here are the subcortical regions, and the far bottom right is the cerebellum. Okay, now let's get to the analysis of this matrix. One striking thing is that proximal regions, so the regions along the diagonal, they tend to be positively correlated. They tend to be. And off diagonal, which is usually the distant regions, they are negatively correlated, which makes roughly sense because proximal regions tend to come from the similar tissue. They will have the same metabolic profile. Distant regions will be more different from each other and will have a negative metabolic correlation. If you look at the distribution of correlation as a function of the interdistance, you're finding this negative trend, the same way you would find this in structural or functional weight too. So I'm going to formalize the first rule of MRSI connectomics. We are maximizing local metabolic similarity or minimizing local variation, the same way we would maximize local wiring in structural connectomics. Okay, what happens if we try to discover the underlying manifold or dimensionally reduce this big matrix and then map it back to the brain? We're ending up with these pictures. Okay, so the color code means metabolic similarity index, which is just a 1D dimensional representation of those 5 metabolites that we saw in the beginning. Similar color means metabolically similar. Okay, we're seeing again this local trend. Regions that are proximal to each other will have more or less the same metabolic profile, with few exceptions. Both hemispheres are completely homotopical. They are symmetrical. The lateral part is very similar to the other lateral part, so they are distant from each other, but still they maintain the same metabolic profile. Also, the occipital lobe and the cerebellum are lying right next to each other, but they are completely different from each other, so there is no point spread function, no blurring of the scanner going on here. And actually, you can model, 10 billion random geometric models, take the best one, and it will only explain about 0.1 correlation with the observed matrix. So proximal connectivity is not enough. We need the second ingredient. If you look closely at these maps, you get a very nice separation between anatomy and functional. Sensorial from motor, higher order from motor sensory. We get an isolation of the limbic system, and cortical from subcortical. There's a very nice separation of each of these regions going on here. And also, if you look at the histogram of these values, it's almost randomly uniform, meaning that it's very rare to find redundancy in these values. There are no regions that will have the same metabolic profile. We get the almost perfect separation of metabolism. Okay, but we already knew this. What's the new thing here? The new thing is that we can inverse map this into metabolic space. Okay, so we now know that the occipital lobe has that particular separation from the rest of the cortex. Why? Because it has more TNA and less choline. We now know that the subcortex is different from the rest of the cortex. Why? Because it has more choline than TNA. And choline and TNA are the metabolites that are going to drive this whole variation, where we're getting a flip somewhere in the frontal regions. And this is the thing that is going to drive the whole pattern that we're seeing here. So formulate the second rule of MRSI connectomics. We're going to minimize redundancy or maximize entropy of these values. Why am I showing this? Let's get a very simple example. Let's imagine a one-dimensional grid of values, perfect randomness, maximal entropy. Values are completely distributed. There is no pattern forming here. But once you implement this local similarity in there, you're starting to see a gradient appearing in the values. But the gradients will be distinct, they will be split in two. Now, once you enforce redundancy again, meaning that there are no two values that are going to be the same, we're getting a one single gradient, a curve appearing in one dimension. This is going to be a bit more complicated in 2 dimension or in 3 dimension because the problem is overdetermined, but this is how it looks in 3 dimension. We're starting to see a variation starting somewhere in the occipital lobe, going to the frontal region, back to the subcortical regions in a nice, smooth, and gradual way. And this whole pattern is mostly driven by the flipping of the TNA over choline ratio. We can map those curves back in 3D, and that's what we get. We get a progression of this metabolic gradient starting somewhere in the occipital lobe, going towards the frontal lobe and ending somewhere in the subcortical regions. And the nice part is about this that it's passing through all the high similarity nodes, the very well connected nodes in the metabolic sense. Okay, now we can formalize this principle, the economy of metabolite distribution rather than the economy of brain networks, where the first rule is minimize local variance or maximize local similarity, and also maximize global diversity or entropy. And so our brain actually finds a sweet spot between a completely random geometric topology and a complete random topology. We are somewhere in the middle. Other people, researchers have found these gradients. Of course, if you have assisted to the workshop in the first day about gradients in different modalities, it's not something new. But the thing that stands out here that we have a completely continuous gradient that goes through all the main regions along the cortex and the subcortex. And the study that replicated the best, that seems the most similar to what we found here, is this one, where you look at the thalamocortical axis and try to combine the structural data with genomic data. Okay, how does this compare with other networks? Structural, not that much. We only get a 0.05 correlation with the structural one. We can even test other metrics, communicability, transmissibility. There is little similarity between the structural and the metabolic one, which is surprising, but also it gives us hope that we are in fact dealing with something completely different that is orthogonal to what you would do in structural MRI. However, if you delineate the structural nodes that are highly connected in the structural sense, of course, well, they tend to have higher MRSI concentrations, which means that, okay, something interesting is going on. They are highly connected in the structural sense because they have high MRSI concentrations. What about cytoarchitecture? Von Economo data set, the big brain data sets, we get a 0.1 with the von Economo. It's not that much, but we get an almost 0.3 correlation with the big brain data sets, which means that metabolic similarity closely aligns with cytoarchitectural similarity. What about genetic co-expression? If you download the Allen Brain data set and map this to the same parcellation that we used, we get a decent 0.46 correlation, which means that, okay, metabolic similarity could be explained in part by genetic similarity. All right, what about future clinical applications? We have outlined as that this TNA over choline ratio is actually important. It's also a marker for neurodegeneration, so we can actually measure this with an MRSI scan. Also, we showed that metabolic content predicts structural connectivity. We might hope that, for example, in early psychosis, this pattern would be disrupted in those particular patients even before we see the disruption in the white matter tracts. And the other interesting thing was this gradient that I showed you before. We might see a disruption in that gradient in early psychosis patients. It looks abstract, but we can always map it back into the metabolic space. So if we see a disruption somewhere in the temporal lobe or the frontal lobe, we know exactly which metabolite disrupted that particular gradient at that particular region. And we actually did this. So I just started doing this patient control analysis. So here on the left, the healthy controls. So you get a nice gradient going from left to right. So this is not Euclidean space, this is just an embedding space, which helps me do the analysis. In healthy controls, this is more or less very nicely actually preserved. So we're starting with violet, going to blue, then red. In CHR, so clinical high-risk patients for psychosis, this pattern is disrupted somewhere in the frontal regions. And we know which nodes have been disrupted. We can actually pinpoint the nodes that are going to disrupt that particular pattern. Saying, for example, over on top there was too much choline. Here at the temporal, maybe, I think it was the temporal region, so the TNA was too low. If we could somehow, push those metabolites up, re-equilibrate that, we can maybe preserve and maintain that pattern. I will close down with this slide. I would like to thank my research group, Paul Klauser, which is the head of the research unit for psychosis, Edgar Silro, right here. Go visit his poster. We were working 1 year straight to do all the pre-processing of this highly noisy data set. We did this with the utmost care. And of course, you can download all of this pre-processing pipeline that we developed over the year on our GitHub. Just scan this QR code. Of course, our 2 publications are on preprint now. So on the left, the Connectomics part, which is the work that I just presented. And if you're more interested in the Voxel-Based analysis, scan this QR code. It just went online a few days ago. And with that, I close off this thing. And I'm looking forward to your questions at the very end of the session. Please post them in the Q&A or keep a note, and we'll try to address them as good as possible. Thank you for your attention.